Hello, everyone. I'm Sai Lakshmi Subramanian, Program Manager of the Seven Bridges Cancer Genomics Cloud. I'm delighted to welcome you all to the third month of the CGC monthly webinar series. Just wanted to introduce the webinar series in case you missed the last two webinars. All the resources about the current webinar, the slides, the recordings, everything's available at the Cancer Genomics Cloud website. So please go to cancergenomicscloud.org/webinars to look at the resources. And just want to let you know that these webinars happen every month on the fourth Wednesday of each month at 2 p.m. Eastern. So please save the date and time. Information about the upcoming webinar will also be posted in the website at the same link. Please use the chat box for asking any questions, or if you have any issues with audio or anything, please use the chat box. And at the end, we'll post a survey link, so please respond to that. With that, I'm very delighted to introduce the speaker of today's webinar, Dr. Xavier Bofill-De Ros. Dr. Bofill-De Ros is a research fellow at the Center for Cancer Research, CCR, at the National Cancer Institute. He is studying the role of microRNA biogenesis in tumors and its applications in translational research. He has a broad range of interests, including cutting-edge research and technology adoption in the pharmaceutical industry. Additionally, Dr. Bofill-De Ros is an avid reader, and he has received several awards and honors. Particularly, I'm very proud to say that he was the recipient of the CGC Collaborative Project Award twice in the past few years. And that has led to at least 3 papers on the work he has performed on the CGC, and a few more publications are in the line. Recently, he was awarded the CCR Excellence and Postdoctoral Transition Award by the National Cancer Institute in 2019. With that, I would hand over the platform to Dr. Bofill-De Ros. Over to you.
Hi for your kind introduction. Okay, so can you see my slides correctly?
Yes. Can you move to full screen presentation mode?
Yes. Oops. What is wrong? Oh, okay. Okay, right now. Perfect. So thank you so much, Sai, for this kind introduction and the invitation to participate in this seminar series. The title for my talk today will be Uncovering Novel Roles of microRNAs in Tumors Using the Cancer Genome Cloud. So as we all know, the central dogma of biology states that DNA encodes for RNA and RNA is translated into proteins. However, during this process, there's like a few exceptions, and one of them is microRNAs. MicroRNAs are this class of small non-coding RNAs, the function of which is basically to downregulate this process by targeting the RNA and promoting a faster decay of the RNA and blocking the translation into proteins. The way microRNAs do that basically is by a partial base pair complementarity between their sequence and the sequence of their target genes. And in particular, in this process, there's an important region known as the seed that basically drives this recognition. Based on this seed recognition, microRNAs have been estimated to regulate nearly 60% of the cellular messenger RNAs in the cell. And they are found to be regulating nearly all biological pathways. More importantly, microRNAs are found in all kingdoms of life, animals, plants, and viral genomes. The talk for today will focus about the role of microRNA biogenesis in tumors. And just to briefly bring the audience into this field, I want to mention that changes in the microRNA expression levels have been linked to tumorigenesis for a long time. And it has been observed that tumors tend to have reduced level of the enzymes that are driving the production of microRNAs, that impair microRNA processing enhances tumorigenesis, and even the relationship between non-oncogenes such as MYC. A little bit more into the detail, also people have observed that there's like hotspot mutations in both enzymes involved in this process, and even there's a specific disease known as Dicer syndrome that that basically leads to an array of pediatric tumors. And also they are found both in germinal line and in somatic cells. And also a little bit more into detail, but there's also cofactors of the microRNA biogenesis pathways, which have also been linked to the generation of certain kinds of tumors. So to go a little bit more into the mechanism of microRNAs, I want to briefly introduce the microRNA biogenesis pathway that starts from transcription from the genome into these long non-coding transcripts known as primary microRNAs. And they have this particular structural feature that they fall into a hairpin-like structure. This hairpin-like structure is then recognized by Drosha and DGCR8, and this complex basically triggers the endonucleolytic cleavage, releasing this hairpin. Then the hairpin is exported into the cytoplasm, where another endonuclease known as dicer makes a second cleavage. And finally, this short duplex of RNA is loaded into Argonaute, where it can lead to the regulation of transcripts that I previously described. When we think about microRNAs, we tend to think always about the canonical sequence that is annotated on most databases. But for those that we work on in the field, we know that there's a great deal of variability on those sequences, and those variations are known as microRNA isoforms or isomirs. And they tend to occur primarily on the 5' end of the microRNA and the 3' end. Collectively, isomirs can represent between 20 and 80% of all microRNAs reads. So given this large abundance of these isoforms, our group have been focused on studying them. And for today's talk, I will be basically going over 3 points. The first of them is the development of computational tools that we have been working together with CGC in order to be able to study them in the context of big data sets of tumors. And the 2 other points that I want to deliver today are related to the origin and function of both 5' and 3' modifications, and a little bit the work that we have been doing on those lines. So without any further delay, I want to basically go over the first point. So the first point basically arises from the fact that a few years ago, or a little bit more than that, the National Cancer Institute basically sponsored this big study known as the Cancer Genome Atlas that contained more than 11,000 patients. And basically, this big data set covered lots of different tumors, and over the years, it has allowed basically to betterly characterize molecularly the basis of cancer, to identify cancer subtypes, and even in some cases to approximate what would be the better therapies for treating them. And our take on this big data set and the way we want to take advantage of this is basically to identify modifications with biological and disease relevance. On the top of that, the NCI have also sponsored similar studies focused on pediatric tumors, and those are basically collected under this also pretty large data set known as Target. So with such a big amount of samples available for us to study the role of microRNAs, we had to come up with a tool that was able to handle this big amount of data. And with that, we came with our approach, which is known as QuagmiR. And the QuagmiR algorithm, basically what it does is basically it focuses on the detection of isoforms by pulling from a motif all the sequences that contain this microRNA motif, and then on a second step, filtering them based on a certain criteria on the 5' end and the 3' end. And then the resulting reads are further analyzed and reported in different report files. The main advantages of this way of analyzing microRNAs is basically that we have the researchers have a full control over the filtering steps, and also it takes into account a little bit what we know from biology about where the modification happens, and we are able to set a different threshold of tolerance for mismatches on the 5' end and the 3' end. And on the top of that, we are able to take advantage of the computational power of having a cloud computing behind. As it has been shown in other seminars from this series, QuagmiR is in the Cancer Genome Cloud, and that basically allows us to put our application in the context or in workflows with other well-established applications. And for instance, here what I'm showing is how easily we can integrate a workflow where we have QuagmiR on the center, but also we can easily basically modify BAM files into FASTQ files, which is the file format that QuagmiR takes. And we can also integrate that with some quality analysis of the reads that we are analyzing in a very simple manner. In order to go a little bit more into the detail and to show the audience how we can basically do very simply the analysis of microRNA isoforms, I want to show here this example of a run that I ran a few days ago. And in there, basically, we have this easy setting where we can basically decide whether if we want to analyze the FASTQ files on a batch mode or together. And for that, basically, my recommendation in terms of how QuagmiR performs, I suggest that the users use the batching only when they have a large amount of files or very big FASTQ files. Otherwise, QuagmiR can very easily handle all the files together. Then the next thing that we have to input to QuagmiR is the FASTQ files and a reference file. The reference file is the only thing that is a little bit particular from QuagmiR, and basically it contains on the first line the name of the microRNA that we are analyzing, and after in a space we have the motif that we are setting to pull the reads, and in the next line basically we have the reference sequence of the microRNA that we are analyzing. And with that, basically, we can include as many microRNAs as we are interested to analyze, or if the user have a particular vital microRNA that they want to include on the top of the host microRNAs, they can do that very easily. For our default setting of motifs, we use a 13 nucleotide sequence on the middle. And the reason for that is because based on previous analysis, we have detected that 13 nucleotides is a relatively optimal setting for capturing most of the microRNAs and having enough specificity. The second element that is important on the setting of the run is to decide what we want to use as edit distances, and that is basically the tolerance the program that will have for mismatches either on the 5' end or the 3' end. And for that, our experience with the program indicates that since most of the changes are happening on the 3' end, we want to give the 3' a little bit bigger number, and we tend to use 2 and 5 for that. So with these very simple settings, we can just basically hit run, and in a few minutes we are going to have the results from the analysis. Here I'm showing that this run that took exactly 8 minutes. And basically, the output files that we want to pay more attention, the first of them is this group summary that basically presents a report of the isoform profile per microRNA. And if we need a little bit of more granular data, the other file that comes pretty handy is the group sequence information. And that basically provides granular information for individual reads. When we publish QuagmiR, we are in collaboration with the colleagues in CGC. We also benchmark our program against other microRNA-specific softwares. And as you can see here from the different collection of synthetic reads that we generated, QuagmiR performed really well in all of them, indicating that basically QuagmiR is, since it's a very tailored program to analyze this kind of isoforms, it performs really well on this context. With that, I want to move to the second point and start showing you, all of you, how we have been using QuagmiR, and in particular, in this point, we are going to talk about 5' modifications. 5' modifications, and as the name indicates, is basically any gain or loss of nucleotides at the 5' end of the microRNA, and basically those isoforms can be generated at 2 points during the biogenesis of microRNAs. The first of them is the miscleavage of the endonuclease known as Drosha. And the second one, obviously, is the miscleavage from the other nuclease known as dicer. And the importance of 5' isomers is basically because this small gain of one or loss of one nucleotide or two at the 5' end basically shift the sequences. So whenever we have a shift of the seed sequence, basically we are like basically in a certain way we are basically like moving the pattern of recognition, and that image is can be easily seen, for instance, in this metaphor of a key. So whenever we have any small 5' shift, we are going to be changing how the targets are being recognized, and that basically triggers that for every 5' isomer, we are going to have the targets corresponding to the canonical sequence, but also the targets responding to this newly generated 5' isoform. So with that, it comes a very critical mechanistic question, which is where to cut, how these endonucleases establish their cleavage site. Previously, people have shown that in the case of Drosha, Drosha works as a ruler that measures the length of this stem and establishes very precisely where to cut. And in the case of dicer, basically dicer relies on the two nucleotide overhangs left by Drosha and the distance from the loop to establish their cleavage. So it is easy to envision that any miscleavage from Drosha will also impact on how dicer recognize the product from this initial cleavage site, and also trigger a secondary miscleavage event. And both events will have also an indirect effect on how Argonaut eventually selects the strand that is being loaded into Argonaut and becomes the mature microRNA. In order to study this phenomena, we decided to take an approach that rather than exploiting mutations designed by us as a researcher, we wanted to explore this naturally occurring case, which is the paralogs of miR9. miR9 is basically encoded from 3 different loci in the genome located in chromosome 1, 5, and 15. And when we started working on that, everybody assumed that each of them was contributing to that pool of miR9 canonical. But interestingly, despite they all encode the same miR9 sequence, each of them have different structures surrounding the mature microRNA. So we wanted to see how Drosha handled those different structures to determine the cleavage site. To study that, basically, we cloned each of them individually, and we were able to express them very easily. And here I'm just showing a northern blot to show that all of them are still functional, and all of them produce mature miR9. However, in order to know precisely the sequence composition of these mature reads, we took advantage of next generation sequencing, and basically we generated libraries for each of them. When we map back the reads back into the primary microRNA that was being expressed, interestingly, what we observed is that miR9-1 had 2 distinct cleavage sites on the 5' end. One on the canonical position with 86% of the reads, and a second one with nearly 15%, one nucleotide downstream. Whether miR9-2 and miR9-3 had a very precise cleavage site only on the canonical position. That indicated that during evolution, miR9-1 has evolved to generate this novel species of microRNA. Next, since we are part of the National Cancer Institute, we wondered if this isoform that we detected for miR9 would have a role in tumorigenesis. And for that, using the Cancer Genome Atlas, we noticed that miR9 is highly abundant on a kind of brain tumors known as low-grade gliomas. Interestingly, when we look at the microRNAs that are highly expressed in low-grade glioma, we observed that miR9 alternative, this isoform that comes from the miscleavage of miR9-1, is the third most abundant microRNA in those tumors. So based on that, we made a prediction of which target RNAs could be regulated by miR9 canonical and which ones would be regulated by the alternative isoform. And in order to validate if this different set of target was taking place in the tumors, we did a comparison between patients that had low levels of miR9 alternative with patients that had high levels of this alternative isoform. And what you can see in this cumulative plot is basically that in black line, you can see that genes that don't have any site for miR9, they have a normal distribution around 0, indicating that half of the genes go up and half of the genes go down between those two groups. But more interestingly, those genes that have either one site or more than one site for miR9 alternative are shifted into the left, indicating that they are downregulated when we have high levels of expression of this miR9 alternative. Some of the genes identified from this RNA-seq analysis of the patients were the 3 that I'm showing right now, and all of them had a really interesting prognostic effect on low-grade glioma patients. That was that high levels of expression of that gene was correlated with poor survival, while low levels of expression had a better outcome. And interestingly, this relation was inverse for the expression of the host gene of miR9-1, where we had that higher expression of this host gene had a better prognosis, which is basically what would be the expected function of the microRNA negatively regulating gene expression. In order to validate these targets, basically we generate reporters, and what we are showing, I'm showing here, is basically that those reporters are specifically downregulated by the expression of miR9 alternative. Another aspect that I'm also interested to explore is the role of this miR9 alternative in the context of healthy cells. And as you can see from this northern blot, miR9 is a microRNA that is highly specific from the brain, and also it's found in most of the brain tissues in high abundance. However, if we analyze previously published data sets, where basically they generated a mouse model that can easily parse out different cell types within the brain, we can see that within the brain there's specific cell types that have high levels of this isoform, whether some others have relatively way lower levels, while the overall level of miR9 keeps constant in all of them, indicating that there could be a differential expression of this miR9 alternative. Also, taking advantage of the advent of single cell sequencing, I analyzed the 10 million cells from 10x Genomics. And basically here, I'm just showing how the different paralogs of miR9 have differential patterns of expression across the different brain cell types that come out from single cell sequencing, indicating that maybe the brains have evolved to use different paralogs for different cell types and in order to regulate different groups of genes. So given this biological importance, we wanted to understand a little bit better how Drosha determines the cleavage site on miR9. And for that, basically, we took a genetic approach where we generated chimeric constructs that combine different elements of the structure of miR9, which are basically the flanking sequences, the lower stem, and the terminal loop. So whenever we generated chimeric constructs introducing elements of the structure of 9.2 into miR9-1, what we observed is that the lower stem of miR9-2, sorry, was the one reducing the most the miscleavage. And on the contrary, when we introduce elements of the structure of miR9 into the structure of 9.2, the lower stem of miR9-1 was the one promoting the highest alternative cleavage. So that basically pointed that there was something special with the lower stem of miR9-1 driving this alternative cleavage. In order to study that, we collaborated with two groups in order to do a biophysical measurement known as a small angle X-ray scattering, which basically measures the 3D structure of naked RNA. And as you can see from these two figures, basically miR9-1 in 3D tends to bend around the lower stem, whether miR9 tends to form the A helix from the RNA that we would expect. So based on these insights, we generated 2 hypothesis-driven mutations that either corrected the asymmetry of the bulge at the lower stem of the microRNA or made a perfectly complementary stem. And in both cases, we were able to highly reduce the alternative cleavage that we saw on the original wild-type sequence. On the contrary, if we generated mutations that were even more prone to 3D distortion, we had an even greater level of alternative cleavage. Next, what we wanted to study is whether this phenomenon of alternative cleavage was also present in other primary microRNAs, and to which extent this alternative cleavage could be explained by our mechanism. And for that, basically, we took advantage of TCGA, but in this case, in particular, we use only normal tissues. And basically, we classified a long list of more than 100 microRNAs based on the precision of their products. So on the lower part of the graph, we can see those microRNAs that consistently among different tissues, they produce mature microRNA products that have a very precise 5' end. Whether on the top we have microRNAs that are more sloppy on their cleavage, and that will lead to different seed sequences. And when we compare the structure of these 2 groups, what we observe is that those that have multiple cleavages tend to have an increased 3D distortion and the presence of flexible motifs. And to validate that, we pick a couple of examples, in particular the miR411 and 10B, and we corrected their lower stem structures. And in both cases, we were able to highly reduce the alternative cleavage. However, all these studies till this point were observational, but in order to generalize our model even more, we took advantage of a data set previously published from the group of David Bertel, where he generated more than 80,000 sequence variants on the top of the primary microRNA structure. In there, by calculating the folding energy of those sequence variants, we were able to observe that those that are less flexible tend to have a more precise cleavage site, whether those that are more flexible or more prone to 3D distortions are more prone to have alternative cleavage. Indeed, to go a little bit more further, we were able to collapse those sequence variants into their common secondary structure, and that basically yield around 1,300 secondary structures. And when we compare all of them to the wild-type sequence, what we got is basically a group of structures that, independent of their nucleotide composition, they are prone to this alternative cleavage. As you can see on the upper right corner of this plot, we have a group of structures that they have a very statistically significant alternative cleavage production. And when we look into a little bit more of detail into those structures, we can see 2 different groups. One of them basically falls into the category of disruption of the lower stem length, which is the mechanism that was previously reported on the literature. And the second group of structures that we detected is basically structures that have big bulges on their lower stem, indicating that those structures are basically a generalization of what we observe for miR9-1. So summarizing this first part of my talk, basically what we observe is that the structure of primary microRNAs is heavily has a high importance on the production of 5' isoforms, and those 5' isoforms can basically provide novel functions for microRNA paralogs. What I'm doing right now is basically going a little bit more deep into into the production of 5' isoforms in tumors, and in there basically I'm studying how mutations on the protein N could also lead to misprocessing of microRNAs. And just to show you very quickly one example of that, what I detected is that certain mutations that are located on the interface between the RNA and the protein are basically involved on the precision of this production, indicating that maybe tumors are exploiting the misprocessing of microRNAs to their own advantage. Another aspect that I feel very interested to keep developing is if the dysregulation of RNA binding proteins that bind nearby the primary microRNA could also have a role in that. And for that, I'm taking advantage of TCGA using a very intensive bioinformatic approach. And also at the same time, I'm trying to reach the same conclusions by generating a biochemical proximal labeling approach. Moving forward to the last point of my talk, I want to talk a little bit about 3' modifications because they are a little bit more abundant. And there I want to talk about our recent paper in the context of microRNA decay. So in order to understand a little bit more 3' modifications, I want to mention that microRNAs are not naked RNAs around the cell, that they are always in the context of this proteins known as argonauts. And basically, the reason why the 3' N is more usually modified and after the maturation is for the fact that the 5' N is deeply buried into a domain of argonaut that protects it very well. However, the 3' N is loosely protected by this domain known as the pass domain. So whenever the microRNA exposes this 3' N, that makes it prone to either losing nucleotides or gaining new nucleotides that are not templated, that originate after the microRNA has been processed. In order to study that, we generated a point mutant of argonaut on 2 residues that we call the F2L3. And this F2L3, basically, it's a mutant that promotes the dislocation of the 3' N of the microRNA from the pass domain. As you can see on this northern blot, where we overexpress 1 microRNA, whenever we have this mutant, the 3' N is very loosely locked into this domain, and that basically leads to this ladder of isoforms that are shorter and longer than the canonical sequence. And from the right panel for this microRNA, you can see that basically the gain of nucleotides is non-templated, indicating that all those events happen after the maturation of the microRNA. Interestingly, if we also do a pull down of endogenous microRNAs from the cell, either from wild-type argonaut or this mutant that have a loose binding of the 3' N, we can see that for all of them, all these 100 microRNAs, in all of them we have an increased percentage of 3' isoforms, indicating the importance of this domain on protecting the 3' N. In order to study that a little bit more in the context of tumors and how the 3' N is protected in tumors, basically we took advantage of the Cancer Genome Atlas, and in there the analysis that I performed, basically it consisted on comparing individual amino acid mutations that changed the residue on the protein N to a big group of all missense mutations or all synonymous mutations, assuming that either all missense mutations and all synonymous mutations will have a little effect on the 3' N modifications. And once I generated those groups, I compared the microRNA-seq datasets, and in particular the 3' N profile of the microRNAs, the trimming and the tailing. And what you can see from the results is that we identified 2 mutations on tumors, in particularly the proline 295 and this arginine 315. And as you can see, similar to the artificial point mutations that we generated on the overexpressed protein, these endogenous mutations uncovered in these TCGA patients had exactly the same phenotype as you saw before. All of them basically lead to a huge increase on 3' isoforms. And if we look at the structure of the protein, we can see that both of them basically locate into the interface between the 3' N and the microRNA. On the contrary, another missense mutation happening also on the pass domain, but on this loop outside of the interface between the microRNA and the protein, have no consistent effect for any microRNA, indicating that we can use the data from TCGA to basically observe this same phenomena. And more interestingly, we find these mutations not only on Argonaut, that is the most abundant Argonaut, but also in Argo1, which is also a very abundant one. And in both cases, we were able to find mutations that show this phenotype. So in order to understand a little bit better what happens when the 3' end is exposed, we generated a mutant that that basically deleted the whole pass domain. And when we do this mutant, what you can see from this northern blot is this extremely long ladder of isoforms that basically extend like even 10 or 20 nucleotides the length of the microRNA. And when we analyze those isoforms by deep sequencing, what we observe is that basically they were constituted by an increased a very important addition of uridine on the 3' end of the microRNA, but not any of the other nucleotides. So this specificity for adding U at the end of the microRNA quickly pointed us that the responsible enzymes could be 3' uridyl transferases, and those enzymes is basically a group of non-canonical polymerases that are known for adding U's or A's at the 3' end of the microRNAs. And in particular, the ones that have been described for adding U's is basically TUT4 and TUT7. So in order to study them, we generated individual knockouts of those two enzymes, TUT4 and TUT7, and also a double knockout of both of them. And as you can see here, when we express the past deleted mutant, in both cases, if we don't have TUT7, we have a huge loss of these long tails. And if we have the double mutant with TUT4 and TUT7, the loss is even more, indicating that TUT7 have the biggest role, but also contributed by TUT4. And once we know that that these two enzymes were the responsible for adding U's, immediately we link that to previously published papers that indicating that TUT4 and TUT7 addition of non-templated U's may make any RNA a substrate of this exonuclease known as dis3L2, which is involved on the degradation from 3' to 5' or of RNAs. So we also generated a dis3L2 knockout mutant, and here there's a lot of lines, so I want to guide you slowly between them. So when we compare past mutants between the wild type and the dis3L2 knockout genotype, we saw an increase of those tails, in good agreement of dis3L2 being the enzyme that removes those microRNAs, but also on the F2L3 mutant that have this small point mutation on the pass domain, we also observe an increase on the tails. However, there was no increase on tailing on AGO wild type, neither on the wild type genotype or the dis3L2 mutant, indicating that in any case we need the exposure of the 3' end to trigger this degradation mechanism based on the coupling of TUTs and dis3L2. So the next question was to identify which microRNAs are endogenously being regulated by this mechanism. And by sequencing the dis3L2 mutant and comparing that to the wild type with no expression of any microRNA at all, what we identified is that there's at least 3 microRNAs, miR7, miR222, and 769, that grow very long tails when we disrupt the dis3L2 activity. And we will in each of them, basically all of them are gaining long tracks of use. And when we validated that by northern blot, what you can see here is that all of them have this long, this kind of a smear or ladder of length, And this length is completely abolished when we on the top of that we have the knockout of TUT4 and TUT7, indicating that the exonuclease needs the priming from TUT4 and TUT7. Then the next question that we try to address is what was the specificity of generating these tails by TUT4 and TUT7? And basically for that, the fact that we identified miR7 clearly pointed out to a mechanism known as target-mediated decay, which is basically when we have a non-coding RNA that have extensive complementarity with a microRNA. When that happens, that basically triggers the dislocation of the 3' end from the microRNA from Argonaut and expose that for modification. So when we analyzed computationally the presence of this kind of binding sites across the 3 microRNAs that we identified, we observed that for all of them, we had a large amount of genes that could promote this dislocation. Whether for the controls that we pick, like miR21, 148A, or miR10B, there was not such targets that could dislocate the microRNA. So summarizing this story, what we identified is that when microRNAs bind to one of those target RNAs that have extensive complementarity, that can trigger different decay mechanisms. And the one that we unveiled is basically the addition of long tracks of use by TUT4 and TUT7, and that being coupled with this 3L2. But also we are currently working in 2 other scenarios where other exonucleases could also trigger the degradation of the microRNA when they expose the 3' end. And lastly, I want to basically go a little bit over another function that we uncover for 3' modifications, which is in the context of target recognition. The idea behind this project arose from what we knew about 5' ends, and as you remember from the first part of my talk, the seed sequence is basically the driver of the recognition. So we were wondering if for those targets that don't have a perfect base pairing on the seed sequence, if the 3' end could be doing a similar role. If the 3' end could be compensating the lack of the seed sequence, and if the 3' end could be driving a loss of function or gain of function, depending of the base pairings. And in this case, we're already thinking that the enzymes involved on this target recognition would most likely be the same TUTs that I just mentioned before. So from reporter assays, what we observe is that basically when we have a target pairing that don't have the seed sequence, if we have an extensive complementarity at the 3' end, we could still have repression, indicating that the 3' end could play a role on that. And more interestingly, when we look the effect of the nucleotides adjacent to this 3' end, we observe that those targets that have an A available to be base paired with a non-templated U were also able to provide repression. Whether those that had a C that doesn't base pair with the U or the A didn't have any repression at all. In order to generalize this model that we observed in the luciferase reporter and that we give the name of Tail-mediated repression, what we analyze is basically cells where we overexpress a microRNA, and we basically divided the genes similar to what I showed before with this cumulative plot where we have a big group of genes that don't have any target at all, and those have like a standard distribution around 50%, half of them going up and half of them going down. Then the next group that we have is the ones that have a canonical site in the red line, and those, as expected, they have repression, indicating that the microRNA is actively repressing them. But more interestingly, we define these other groups of targets that are under this Tail-mediated repression, and those per se have a relatively more moderate repression, but in combination with the canonical sites, basically they even enhance more the repressive activity of microRNAs. Interestingly, when we did exactly the same experiment in the context of the knockout of TUT4 and TUT7, the enzymes involved in modifying the 3' end of the microRNA, what we see is that the tumor or the tail-mediated repression is completely gone, and the group of genes that were even more repressed than the canonical ones on the previous plot, they basically behave as canonical microRNAs. And that basically, at least for this particular microRNA, proved the idea that the 3' end could be involved on modulating target recognition. And that is a very interesting idea, because till that point, everybody believed that microRNAs were, if they were there, they would be repressing. But what we are showing with these modifications is that microRNAs could be selectively recognizing or not recognizing certain kinds of targets depending on this modification on the 3' end, indicating that their activity could be under regulation. In order to generalize this model to other microRNAs, what we are currently doing is basically performing a kind of experiment known as AGOZ CLIP. And basically, AGOZ CLIP is a technique that, similar to other CLIP approaches, basically it cross-links the RNA to the protein. But in the particular case of the microRNAs, there's an extra step that basically consists on the ligation of the 3' end of the microRNA to the 5' end of their target. So in this way, whenever we read those chimeric reads by deep sequencing, we can identify precisely which microRNA is recognizing which target. And the idea behind that is to identify in the context of wild-type cells which targets are being recognized in this mixed population of microRNAs that are modified or unmodified by TUTs, and also to compare that in the context of the TUT knockout, where there's no modification at all on the 3' end. And just to show you a little bit of preliminary data that we have on this end, what we are doing is basically analyzing all those chimeric reads using RNA folding programs. And what we can see here is an example on how a microRNA is recognizing a target. So in red, you have, and with this dot bracket notation, you have the annotation of which nucleotides are being best paired between the microRNA and the target. So in gray, I'm going to show nucleotides that for each chimeric read are paired, or which nucleotides are unpaired. So this pretty busy plot, what it is indicating is on a list of thousands and thousands of chimeric reads, which nucleotides are being involved on the target recognition. As you can see, the nucleotide 2 to 8, which constitute the seed, is mostly present on all chimeric reads, but only on a fraction of them is the only nucleotides being base pair. On the vast majority, what we see is that there's an extensive base pairing on the 3' end, indicating that all those targets could be subjected to regulation. And we see that not only for miR148B, but we see that for many other microRNAs, miR7 and miR20. And what we are basically looking right now is to see, as I was mentioning, if this 3' pairing shows any difference between the context of wild-type cells or not, indicating if these extra nucleotides could play a more general role on target regulation. With that, I want to basically go a little bit over our model. So going back to the beginning, we have this canonical model of microRNAs being driven by the seed sequence. But what we are proposing right now is basically that microRNAs can have like a gradient of dependency on the seed, being some microRNAs heavily dependent on the seed sequence, but in some others, we could have a more stronger effect from the 3' end. And in particularly on those cases, those targets could be under regulation of this family of microRNAs, of this family of non-canonical polymerases known as TUTs. And just as a teaser for thought, here I'm showing a couple of examples where we see that in tumors, the 3' end of microRNAs is heavily modified, and that is different from normal cells, indicating that in tumors we could still have the same level of expression of the microRNA, but their activity could be modified by these additions, by these gains or losses of nucleotides at the 3' end. So with that, I want to summarize the science that I have been presenting today, where I show that there's a role of RNA structure on microRNA processing, and that basically have a big impact on the role of the seed and on the definition of the 5' end. And in the last part of my talk, I have shown two new functions of the 3' end modifications, one of them being on the turnover of microRNAs by coupling these extensions of oligo-uridylation with exonucleases, and how the addition of single use could also provide a novel mechanism for base pairing between microRNAs and targets, and that provides a novel mechanism for regulating target recognition. And in a little bit bigger context, and since I'm presenting for the Cancer Genome Cloud, I also want to give this take-home message that we, or this idea, that basically in a lot of cases we are able to combine like different data sets, and we feel very interested on this approach that we have taken on these different papers, where we combine deep mechanistic insights of the biology of microRNAs, and basically we are able to translate our findings into big data sets from the Cancer Genome Atlas, with the idea that molecular insights can basically teach us about tumor biology, but at the same time, tumor biology can give us insights on novel mechanisms that we haven't looked before. And with that, I want to thank all my colleagues in the GU Lab, all our collaborations that have been participating in these projects, and the colleagues and friends in 7 Bridges Genomics that have also provided help and support during this time. And with that, I'm very happy to take any questions.
Thank you so much, Dr. Bofill-De Ros. This was an excellent presentation. I really liked your final slide, which told how molecular mechanisms lead to better insights in tumor biology, but it's also the reverse. And I'm extremely impressed with how you have harnessed the power of the cloud in the Cancer Genomics Cloud to uncover a lot of mysteries. There's more to learn, more to uncover, but there's so much that has been done, and kudos to your team. So with that, I would like to read out the questions. So one of our speakers have asked, which target prediction algorithms did you use for your analysis of targets of microRNA isoforms?
Okay, yes, that's a very good question. For the canonical sites, we relied on the gold standard in the field, which is target scan. And for our own targets, depending on the modification, we try to implement a similar algorithm that relies on looking for on evolutionary conserved nucleotides base pairing on the 3' end, and also integrating a little bit the local accessibility of the target sites. And we did that basically using RNA fold and looking to those context sites that allow for an easy accessibility of the microRNA.
Great, thank you. That was great. So if others have any questions, please post in the chat box. We had another user who had requested the recording of the webinar. So I just want to mention that it will be posted in the website along with the slide deck. So after the webinar, maybe in the next 2 days, please be on the lookout. You will be able to access this. I do not see any other questions. So I wanted to check with you on, so you have developed your own tools within the, and released them within the platform. And also, I'm pretty sure you've used other tools within the CGC. So can you talk a little bit about, how is the experience using the cloud, specifically the CGC, for testing and benchmarking your algorithm that you developed?
Yes, actually, that was a little bit my idea when I put together this talk. So basically, in all these projects, we have, there's our core expertise, which is microRNA isoforms. But the beauty of each of those studies is that we have been able to take advantage of TCGA and target, in the sense that for the same samples, you have for all of them, like you have like whole genome sequencing, you have RNA-seq. So what we have been doing is basically combining total RNA-seq expression in order to see how genes go up or genes go down in relation to the microRNAs. And for instance, in the Nature Communications paper, what we did is basically to look at whole genome sequencing or exome sequencing, identify which samples have those interesting mutations, and be able to analyze the effect of those mutations on the domain that we are studying by looking at the microRNA-seq datasets. So I think the Cancer Genome Cloud had been very key in order to have all this data together, to generate groups with the samples that we were interested, and being able to analyze them in a pretty high throughput manner with hundreds of samples at a time, and being able to generate so easily the groups, and also being able to reproduce the analysis with collaborators. Yes,
that's great, and that's exactly the outcome we wanted. And we want our researchers to use the cloud in that manner. So thanks again. I do not see any more questions, but you can find Dr. Bofill-De Ros's email here in the last slide, so you can email him if you have any questions about his research. With that, thank you again for presenting your impressive work here. I just wanted to wrap up with a couple of logistical things. So if you have any questions about using the CGC, please contact the support. The email is provided here. And you can also visit the Knowledge Center. And additionally, we have office hours every week on Thursdays at 2 different times. So you will find the information to log into the office hours in our website. And lastly, I'm very delighted to announce the speakers for our next month's webinar, which is going to happen on the 28th of April. That's the 4th Wednesday of April. We'll have Dr. Vesna Pajic and Nevena Vukojic from Seven Bridges. So they're going to be talking about true multi-omics analysis. So Dr. Bofill-De Ros just talked about how it was easy to combine mRNA or microRNA datasets, RNA-seq and microRNA-seq within the cloud. We're going to take it to the next step to bring in some proteomics data from CPTAC and show you how you can use the CGC for breast cancer multi-omics analysis. So please be on the lookout for the registration. It will be up very soon. I'm hoping to see you all at the next month's webinar. With that, I would like to thank all of you for joining us for today's session. Thank you. Bye.
Bye.